Alright, we are now ready to enter into actual mitosis, the actual division of the nucleus and ultimately of the cell itself. We have gone through from the beginning of the cell's life, where it's just formed, and through G1, where it is basically just becoming a larger cell, then S, where it is replicating exactly its genetic material, so that it will now have 2 copies of all its genetic information, so that it may give each of its offspring cells an exact copy of all the instructions each cell will need to do its job. And then the G2 stage, where it is basically making final preparations for what we're about to talk about right now, mitosis. So let's talk about this. Hopefully you'll remember this from earlier years in biology. Let's see. Alrighty. Alright. Replicated the genetic material. Now, basically mitosis is the act, indeed, of divvying up the one complete set of genes to this offspring and one complete set of genes to this offspring, so that they each have everything they need. Okay? Mitosis really is quite specifically talking about the division of the genetic material, okay, the nucleus, the genes. Then we'll divide the cell after we've done with that division of the genes, that ever important division of the genes. Okay, so we are dividing the chromosomes between 2 daughters. There are 4 stages in which we do that, and you will remember, I hope, prophase, metaphase, anaphase, and telophase. If you can remember PMAT, okay, you'll always know the order of those. And all of those 4 stages, about which we are going to go in tremendous detail, all of them fit into this tiny slice of the cellular life pie. Okay, so please understand that in the great scheme of things, this is really a very small part of the cell's life, even though we take a long time to talk about it. Now, why do cells divide by mitosis? Okay, well, we started touching on this back when we talked about skin at the very beginning. We have skin, we're always losing our skin, we want to make more skin. We want to make exact copies of all our skin cells so that we have skin to replace our skin that comes off. So, we're making exact copies. Now, some organisms reproduce, like make new individuals by making exact copies. This amoeba is doing mitosis to make a baby amoeba. This is a single-celled organism. It is reproducing. It is making new organisms using mitosis. Okay, so asexual reproduction, okay, is often achieved in eukaryotes by mitosis. Bacteria can't do mitosis. Bacteria do something different called binary fission, because bacteria are fundamentally different. They have no nucleus, so they don't do the same things that nucleated cells do. But little single-celled amoeba, they reproduce. I gotta make babies. There you go. Mitosis, one-celled organisms. Now, us multi-cell organisms, however, like maybe this was you as a zygote, and then you were 2 cells, and then you were 4 cells, and then you might become 8 cells, and so on. You're growing by mitosis. You're still growing by mitosis. Okay, so from one zygote to a multicellular trillions of cell organism, all of that happens by mitosis. Okay, also, if you cut yourself, hopefully you will not walk around with a gaping hole in your body forever. Tissue will grow and heal that hole. In this starfish, we can see that this one arm that was ripped off of the starfish actually has given rise to a whole new rest of the starfish. Pretty amazing. All of that is achieved by mitosis. If you've ever seen a lizard, a skink that has intentionally lost its tail to try to avoid being eaten, that tail will grow back by mitosis. So we replace cells that are worn away or are lost or injured or damaged by mitosis. Okay? So the main thing to remember here, really important, is our goal is identical cells to the parent. We want clones. We want clones. We want no differences. We want true, reliable copying of cells in the case of mitosis. All right? Now, we can't really talk about dividing the nucleus, dividing the genes, without talking about the tools, the toolkit the cell has to divide those genes. And the tools the cell uses are cytoskeletal fibers. A skeleton for us means a support structure, and also structures we use to move. If it weren't for your bones, your muscles could twitch all day, but you'd just be this quivering mass of jello on the floor. It's your bones that allow your muscles to work against something and move. Well, a cytoskeleton is basically the same. The cytoskeleton is basically protein fibers that act to move the cell or give the cell support so that it can have a definite structure, a definite shape, or certain organelles can have a definite shape, or the cell can move in certain ways. So cytoskeleton fibers are going to be very important in cell divisions. Okay? The cytoskeletal fibers are important in mitosis because first they move the chromosomes. They are going to move the chromosomes around, organize them, and then those pieces of DNA are going to be separated properly using cytoskeletal fibers. And then ultimately the cytoskeleton is going to actually pinch the cell or cut the cell into two halves so that we have two new cells. Okay? There are 3 types of cytoskeletal fibers. All 3 have one major thing in common, and that is what they are made of. They are all made of protein. Okay, and you should know that. The 3 types are called intermediate filaments, microfilaments, and microtubules. One of these you already know all about, and that's this one, actin, that we learned about in muscle cells. Okay, same thing. Microfilaments are actin fibers, and they work with myosin to contract things. Okay, so we'll talk about that, but you already know all about microfilaments, so really we only need to learn 2 other things. And intermediate filaments are easy. They don't really do much in mitosis. In fact, they don't actually do a whole lot of activity. They're kind of permanent. They're like scaffolding, if you will. They're made out of a particular protein called keratin, which is the same stuff your skin and fingernails and hair is made out of. And it's basically a support structure. They can anchor organelles into position. When the shape is important for something, intermediate filaments can make that shape kind of a permanent thing, if you will. Microfilaments are solid rods made out of actin. And if you'll remember, we talked about actin molecules being like little pearls, and an actin filament is a strand, double twisted strand of those pearls. They do very well at withstanding pulling forces, which, if you'll remember the muscle contraction, they get pulled. And they help not only to support the cell, but also to change the cell's shape. Okay? And you need to know this term. It is responsible for the cleavage furrow at the end of cell division, and that is where the cell pinches in two. We will see this later, so if you're like, I don't understand that, don't worry, you will at the end. Okay? Microtubules are the one that's really, really important for cell division. So I'm going to star this one. In cell division, this is very important. These are the guys that are going to move the chromosomes. They are straight rods. They are hollow, unlike actin. They are made of, conveniently, a protein called tubulin. So microtubules are made of tubulin. Okay? It's just a protein. So we've got three proteins we know about: keratin and intermediate filaments in your fingernails and your hair, tubulin and microtubules, and the actin and the microfilaments. Okay. Now, these microtubules are interesting because they move things by actually growing themselves and shrinking themselves. Tubulin is a little modular protein, kind of like actin, but it's little tubes like this, and each of those would be a molecule of tubulin. And if you want to make a microtubule longer, you just string more tubulin together like that. But if you want to make a microtubule shorter, you get rid of one, 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 until you've made it as short as you want. That's how they get longer and shorter, by physically adding Legos or taking Legos away from the entire strand. Okay. Alright, so this is a real key: disassembly and reassembly. They can become long and then they can come apart and be reassembled somewhere else. They can provide shape and support, but most importantly, they provide almost like little tracks where little things called motor proteins actually walk along these tracks and pull chromosomes or vesicles or whatever needs to be moved along this path in the cell. Okay, but of course the second bullet is the one we're going to talk about now. They'll move the chromosomes during cell division. I'll show you animation later that hopefully will make this very real for you. You need to know these words because you may see them: centrosomes and centrioles. Both of these have to do with the microtubules that are going to move the chromosomes around. So what we see here in this diagram are the big dark things here. Those are the actual chromosomes, but these little fibers here and here, these are the fibers that are going to pull the chromosomes and move them around. Okay, well, these fibers arise from a region called the centrosome, and so this area here would be the centrosome from which these fibers grow. And it is thought that these structures called centrioles help organize these things, and these are called spindle fibers that actually connect to the chromosomes. And it's thought that these centrioles actually organize those and help them to do what they're supposed to do. Interestingly, only animal cells have centrioles. Plant cells have spindle fibers and centrosomes, but only animal cells have centrioles. So it's really not clear exactly what they do, but just so you're aware of their existence and that only animal cells have them. Now, an overview of the cell cycle. We need to remember our stages. Okay, interphase. Remember, the chromosomes are thin spaghetti. This is an animal cell, so we can see the centrioles and their centrosomes off to the side outside the nuclear membrane. Then we have prophase. Don't worry about this. This is something your textbook threw in, and it's everybody's like, what? Don't worry about it. Metaphase, anaphase, and telophase. Okay, so, and then we've got something that is actually its own event outside of actual mitosis, and that's the division of the cytoplasm itself, and that's called cytokinesis, and we'll talk about that at the end. Okay, so let's talk about what's happening quickly in each of these stages. Prophase, those long stringy chromosomes that look like a plate of spaghetti get thicker and shorter. They condense, and that's so that we can separate them. It's not all tangly. We can separate them more easily. Okay, and they become visible as individual chromosomes if you were looking at them under a microscope. Okay, we can actually see that these chromosomes are made up of 2 chromatids. Okay. Now, additionally, in prophase, this spindle is going to begin organizing itself, so the centrioles and centrosomes are going to move to opposite poles in an animal cell, at least. Remember, plant cells don't have centrioles. And the spindle will begin to form. This is just flat wrong. These are not part of that, so my bad. I did not check this PowerPoint over before I showed it, at least not well enough. I mean, anyway. The spindle is going to be orchestrating the movement of the chromosomes. Okay. Additionally, this nuclear membrane is going to begin to disintegrate, and we're going to talk about that hopefully. Okay, there we go. The nuclear membrane and the nucleolus, which we'll talk about later, region in the nucleus, all of this is going to break down, and this is going to make it easier for these chromosomes to organize themselves the way they need to be organized. Okay, now, as far as I'm concerned, this is still part of prophase, and I think as far as most AP teachers are concerned, this is still going to be part of prophase. The spindle fibers that have been created are actually going to attach to the centromere region, but what they're actually attaching to are little structures coming off of the centromere region here called kinetochores. Okay, so the spindle fibers will extend out and attach to these kinetochores. Okay, well, that's pretty much all you need to know. So now we are connecting the chromosomes to essentially the centrioles and the centrosomes. Okay, and now the chromosomes are going to start to organize themselves. Okay, and that's just detail on the kinetochore. If you see that, that is the structure at the centromere where we're going to have little proteins that the spindle fibers cling on to. Now what we're seeing here are chromosomes that would probably be in this very late prophase into metaphase stage. They're very, very thick. We can tell they're definitely double-stranded structures. Okay, so now at metaphase, what's going to happen is the spindle is going to act to organize these chromosomes so that their centromeres line up along the equator, or what is called the metaphase plate. It's kind of an imaginary line, but it's right along the center of the cell. Okay, so that is very important. They are precisely aligning during metaphase along the center of the cell, along a place called the metaphase, meta means middle, metaphase plate. The spindle fibers make sure they are aligned properly. Okay, now, this is just a nice diagram so that you can see the arrangement, but we've pretty much already gone over that, so I'm not going to belabor it. Anaphase. Now, during anaphase, the spindle fibers begin to shorten. Actually, some spindle fibers begin to grow. These actually grow and push against each other so that the cell is actually getting elongated and stretched. But the spindle that is actually attached to the chromosomes, this fiber is actually going to shorten. So I told you about the microtubules being made of little tubulin. Well, they would begin to shorten. They'd get rid of some. And these chromosomes, because this fiber is shortening and this fiber is shortening, are going to get pulled apart. Okay? So they are moving to opposite poles. Here's the North Pole and the South Pole. Please don't really call them that. But we're going to pull them apart at the centromeres, and I will show you the motor proteins that actually walk them away in just a minute. I don't know why they keep doing that. Not actin myosin. Alright. This does require energy. This is worth talking about. So the mitochondria in the cell would be busy producing energy for all this movement to occur. And the cell is getting kind of stretched and the poles are getting farther apart. Okay? Now, we don't really need all this detail. Now, I do like this diagram here. This one chromosome made of 2 chromatids, 2 identical pieces of DNA. In anaphase is double-stranded, but it is going to go to being single-stranded. Basically, it is now 2 independent chromosomes. Okay? These, when they were joined, were called chromatids. When they separate, we go back to calling each one of them a chromosome. Again, it is a single-stranded chromosome, not a double-stranded one now, each of them. Alright, after anaphase we hit telophase. The chromosomes get to the opposite ends of the cell to where they were going. Then what we're going to get are 2 new nuclei forming. In other words, the nuclear membrane that had disappeared is going to begin to come back to surround those DNA molecules, and it's going to come back on both sides. Okay, the nucleolus, we see this beginning to reform here. Okay, we'll talk about what that does later. The chromosomes are going to start to get thin and stringy again. You won't be able to see them anymore. The spindle fibers are going to start to go away, and basically we're putting the cell back the way it was during interphase. The only thing left to do is to actually split the cytoplasm itself. So cytokinesis, the splitting of the cytoplasm in the cell membrane, will begin here. This is a nice diagram showing the cleavage furrow that we mentioned earlier. This is a cell entering mitosis. Actually, it's completed mitosis. It's entering cytokinesis. And what's happening is actin fibers, just like in muscle cells, are interacting with myosin fibers, and the cell is actually being contracted along this line and pinched inward, and the whole thing is just going to slice in two. One newly formed nucleus is over here, one newly formed nucleus is over there, and we're going to have 2 new cells. Now, this is mitosis happening in an animal. You remember what a blastula is. It's that ball of cells. You start with a zygote and you divide, you divide, you divide, you divide, and you end up with well, not big, but a little ball of cells. This is a blastula of a type of fish called a whitefish. Why would we look in a blastula to find cells in mitosis to show you in a picture? Because of course there's lots of mitosis happening in a blastula. That's what a blastula is all about. It's mitosis happening to create more and more cells. So it's a really good place, and it's a good connection to make, that if we want to see cells in mitosis, we might look in a little embryo, because there's lots of cells dividing there. Here you can see the cell at interphase. Please notice this nucleus is very nondescript. In prophase, we are starting to see the chromosomes get more defined. In metaphase, we are seeing them line up along this metaphase plate. They look very heavy and dark. We can also see the spindle here, the centrosome area here. We cannot see any centrioles, however. Now, this calls this anaphase. To me, this looks a whole lot like metaphase. This is kind of a late anaphase. They call it early telophase, whatever. This, if it were later anaphase, what you would see is these more stretched out. I hope I can show you a better image of an anaphase later on than either of these. But early telophase, we are starting to get 2 nuclei almost to the point where they're reforming. And this is late telophase, where the chromosomes are actually starting to get indistinguishable again, and you can see where we've got a new cell membrane formed in between. Um, in plants, cytokinesis is different because you've got that cell wall to deal with. The cell membrane doesn't just pinch in. In fact, what happens is we get new fibers laid down along something called a plate, and you can kind of see this forming here. So we get a cell plate formed in between. There is no pinching in, so to speak. It's a different process. We would not have those microfilaments working in that case. Now, in a plant cell, we are seeing interphase, indistinguishable chromosomes. Prophase, they're becoming more obvious. Metaphase, they're lining up. This is a true metaphase right here. You can really see the line. This is an excellent anaphase. You can see them being pulled apart. And an excellent telophase where we're seeing the beginnings of two new cells. Okay? Now, this is showing you what you might see if you looked in one of our microscopes and you looked in the tip of an onion root. Why on earth would you want to do that? Again, because it's a good place to see mitosis. Plants grow at the tips of their shoots and the tips of their roots. If I'm a plant, I'm going to grow at the tips of my fingers and the tips of my toes. So if we were to look at the tip of one of those toes, the tip of a root, for example, we would find lots of cells in different stages of cell division. It's a good place to look, just like the blastula was a good place to look. And so we might find a cell right there. What stage would you say that was in? I'd say anaphase. If I'm looking here, I see a cell, and it looks like it's got just a bunch of gray. There's a nucleolus clearly formed, a clear nuclear membrane. I'd say that was an interphase myself. Here's a cell. What would you say that was in? Looks like they might be lined up in the middle to me. I would think that was a metaphase, perhaps. Maybe this one here. I'm thinking probably prophase. Sometimes they're on the border and you can't tell, but I'll get you to do a lab activity where you'll be looking at some cells and try to determine what stage they are in. Okay? And just a quickie note, it is thought that mitosis probably evolved from the type of cell division that bacteria or prokaryotes do, called binary fission. Binary fission is not mitosis at all. It is not. Bacteria have a single chromosome that's in the shape of a ring. They have no nucleus. It's very different from what a eukaryote has, and so how the cell divides is also very different. Basically, in a bacteria, you copy the chromosome that's in a ring. There you go, you got your two copies, and then you cut that cell in half, you're done. Mitosis is much more complex than that, but it is thought that it originated from this, and scientists have kind of believe, looking at the different kingdoms of organisms and different examples, they can see stepwise how the evolution of that to full eukaryote animal mitosis might have occurred. We won't go into that detail, just want you to understand there is some understanding of how mitosis could have evolved from what bacteria do, which is binary fission. Alright, that's it. Thanks for watching. I know it's long, but it is very important. Happy trails to you. See you later. Bye.